Welcome students. In this section, we are going to discuss about concept of mutation. So let us try to understand what is mutation. So mutation was the term coined by Hugo de Vries in 1901. He had coined this word while he was working on a plant called as Oinothera lamarckiana, which is also called as evening primrose plant. So let us define what is mutation. There are different definitions for mutations. The first one is like this. Mutation is a phenomenon which results in alteration of DNA sequences and consequently results in changes in the genotype and phenotype of an organism. That is, remember, genes are present on chromosomes. Genes are made up of DNA. So now, if the DNA sequences are made up of nucleotides, nucleotides in turn contain nitrogenous bases. So therefore, if there is alteration in this DNA sequences, if there is change in arrangement of nitrogenous bases on the DNA, this is going to induce change in gene. So this change in gene is going to affect the phenotypic expression. What is phenotypic expression? That is, whatever character is expressed in an individual irrespective of genotype. So therefore, mutation can be called as changes in the genotype which ultimately affect the phenotype of an organism. Or in simple term, the mutation can be defined as sudden heritable change in genes of an organism. So instead of defining in a big manner, this is a very simple way to define mutation. Or you have got one more definition, a heritable change in structure of gene or chromosome or change in chromosome. So already I have told you genes are present on chromosome. So if the sequence of gene is altered, automatically the structure of chromosome changes. So this change in chromosome can bring about difference, or it can try to bring about difference in phenotypic expression. And very important thing is, this character of change in gene should be heritable. What is heritable? Transfer of character from one generation to another. Whatever change has occurred in the gene, that should be transferred to the next generation. So such a phenomenon is called as mutation. Now what is the benefit of mutation? The most important benefit of mutation or significance of mutation is it is going to induce variation. So what is variation? Changes. Changes with respect to character. So already we have studied that genetic recombination or sexual reproduction is going to bring about variation mainly because of crossing over. In the similar manner, change in sequence of gene or mutation can also lead for variation. Thus, variation in turn leads for evolution of new species. Hence, mutation is considered as one of the raw material for evolution of new species. How? By inducing variation. So hence the greatest benefit of mutation is to induce variation. So now let us come for about types of mutation. Mutations are categorized into two types. What are they? The first one is called as the gene mutation, or this is also known as point mutation. Second one is the chromosomal mutation. So let us try to understand what is gene mutation and how it is going to be caused. Gene mutation. A change in structure of gene is called gene mutation. See, already I have told you gene is present on chromosome. Genes is made up of DNA. DNA chemically comprises of nitrogenous bases. So usually in DNA, the nitrogenous bases are arranged in specific manner. If this arrangement is altered, automatically the gene structure changes. So this structure or change in gene is what we call it as gene mutation. So therefore here the change is confined only to the gene, a particular gene. So now how does this is going to happen? As I told you, the gene mutation is mainly due to change in sequence of arrangement of nucleotides in DNA molecule. Already I told you what are nucleotides. Nucleotides are made up of nitrogenous bases, sugar and phosphate, and here the main thing is the nitrogenous base arrangement in nucleotides which form DNA is going to alter. So this change in sequence of nucleotide is one of the important reason for gene mutation. Okay, what causes changes in arrangement of nucleotide? That occurs mainly due to two important aspect or concept, that is addition. A nucleotide may be added, or deletion. A nucleotide may be removed. So therefore, addition or deletion of nucleotides, or in simple term nitrogenous bases, is going to bring about gene mutation. So here how many nucleotides or how many nitrogenous bases are going to be added? There are chances one nitrogenous base may be added, 2 or 3. Likewise deletion, 1, 2 or 3 nitrogenous bases may be removed. So therefore this addition or deletion changes the sequences of nitrogenous bases. So this is going to bring about change in coding sequence for amino acid. So therefore that is what we call it as frame shift mutation. That is addition or deletion of nucleotide changes the reading frame of DNA. Hence this gene mutation brings about what we call it as frame shift mutation. So the best example is sickle cell anemia. We will be studying about this when we try to study about Mendelian disorder. So in case of sickle cell anemia is a disorder mainly caused due to change in structure of hemoglobin. You know, hemoglobin is dumbbell shape, but in case of a person who is suffering from sickle cell anemia, it is going to get altered to sickle shape. So why? That is because of change in what we can call new nitrogenous base sequence. So which in turn changes the coding nature of amino acid. So about that we will see in the next part, that is genetic disorder. So at present the best example for point mutation or gene mutation is sickle cell anemia. So now let us go for the second type of mutation, that is chromosomal mutation. Change in chromosome causes chromosomal mutation. So again this is caused due to deletion or addition of a segment of DNA. Already I have told you chromosome contains DNA. So therefore a part of DNA can be lost or a part of DNA can be added. That is during crossing over. When there is exchange of chromosome between two non-sister chromatids, sometimes extra segment of DNA can be added, or the instead of joining, the DNA segment may be deleted, or there may be increase or decrease in number of chromosome. I will give you the example for deletion of segment of DNA. See, for example, you have got a syndrome called as Cry-do-Cat syndrome. In this, a part of segment of chromosome number 5 will be deleted. So that results in Cry-do-Cat syndrome. Likewise, increase or decrease in number of chromosome. For example, there are many chromosomal disorders like what we can call Down syndrome, Turner syndrome, Klinefelter syndrome. In Down syndrome and Klinefelter syndrome, the number of chromosome increases, whereas in Turner syndrome, the number of chromosome decreases. So therefore, thus we can tell that chromosomal mutation may be due to loss of segment or addition of segment of DNA, or it may be increase in number of chromosome or decrease in number of chromosome. So what is the impact of chromosomal mutation? Chromosomal mutations are known to cause chromosomal aberration, that is chromosomal disorder. Few to list out, as I told you: Turner syndrome, Down syndrome, Klinefelter syndrome, Cry-do-Cat syndrome. So these are chromosomal aberrations. And Other thing is cancer. So chromosomal aberrations are more commonly seen in cancer cell. So this is with regarding to the two types of mutations. So now let us try to understand how this mutation is brought about. That is mutagens. What are mutagens? Mutagens are defined as chemical or physical factors or even biological factors that induce mutations are referred as mutagens, or in simple terms, substances that induce mutations are called as mutagens. So these mutagens are depending upon whether they are chemical, physical or biological. You can classify them into three categories. Physical mutagens, what are they? For example, ionizing radiations like UV radiation, X-rays, gamma rays, alpha particles are known to act as physical mutagens because they cause DNA breakage and other damages in a gene. So therefore they induce mutation. So hence UV radiation, X-ray, gamma rays, etc. they are called as physical mutagens. Second one is chemical mutagens. There are certain chemicals, bromine, sodium azide, soralin, benzene, etc. Even some of our food items or coloring reagent, coloring particles what we add for food or tasty material which are going to be added for food, they are also known to induce mutation. So therefore they can be considered as chemical mutagens. The third category is biological mutagen. That is, certain living components are known to induce mutation when they enter into living organism. For example, transposon, that is a small segment of a DNA introduced into the organism, or it may be viruses or bacteria can also induce mutation. So therefore such living components which induce mutations are called as biological mutagens. So this is with regarding to mutation.